Silage as a sustainable livestock feed and its benefits and challenges. As we know that the basal diet of any adult dairy animal comprises of concentrate, green fodders and dry fodders. That is, the basic diet is having the concentrate part as well as the roughage part. And in that roughage part, we had the green fodders and dry fodders. So among these, you know that this green fodder plays a major role in the production itself and the health of the livestock. Why? Because this green fodder is more palatable to the animals. It is easily digestible, and it also exert a cooling effect to the animal, as well as it is having a very laxative effect. And overall, it is quite economic to feed the green fodder to the livestock. So as we know that this green fodder is very important in the diet of the animal, and of course, when we are feeding a concentrate to the animal, it will definitely be is going to give the greatest production per unit intake, but it is not very economical, especially in our country, in the tropical countries. And I can say the importance of this green fodder in the diet of the animal can be expressed in a way that if an animal is yielding about 5 to 6 liters of the milk, and if a farmer is able to provide the green fodder in the right quantity or in the optimum with optimum quality, then without supplementing any extra concentrate even, the animal can sustain this 5 to 6 liters of the milk without having any sort of concentrate. So you can analyze the potential of the green fodder, provided it is fed in an optimum quality. That means the quantity as well as the quality of the green fodder should be sufficient enough to hold the production. So every farmer should understand the importance of the green fodder as well as how much this green fodder is required to the animal. Because we should know that how much we require that this green fodder is required for feeding, right? So here this slide gives you the idea about the fodder requirement of the animal. So we can see that this green fodder requirement is about 8 to 10% of the body weight. So say for an example, if an adult dairy cattle of 400 kg is there, so the green fodder requirement is around 32 to 40 kgs. And see if we are managing a dairy which is having a 10 cows or 10 cows with 5 calves, then how much will be the green fodder requirement for a year? So we can calculate like that way, that if per day we are feeding 35 kgs of the green fodder to the cattle, and for 365 days it will be round about 128 tons, and for the 5 calves, and we are feeding at the rate of 20 kg per day for 300 days, it will be around about 30 tons. So approximately the requirement of the green fodder is 150 tons per year for a dairy that is having a 10 adult cows and 5 calves. So this much is the amount of the fodder required to the animal. So it's clear that the green fodder holds a specific role in the diet of the animal. But still, you have seen that what is the amount of the green fodder required. But there is a demand for some sustainable feed alternatives. Why? Because we are well understanding that this livestock sector is having a significant role in controlling the livelihoods, the food security, as well as the national economy. With the increase in the demand of animal products, there is a simultaneously there is an increase in the demand of a quality feed also. And the traditional reliance on the green fodder and the crop residues is not sufficient enough because of several factors, there is a shrinking land availability for the fodder cultivation. That is from last 2 decades especially, we can say that 4.4% area of the total cultivable area is under the fodder production only. And this figure is not even rising, and we cannot suppose, also we cannot even think that there can be any further expansion of this figure, because there is a extra pressure on the land for the cash crops and the food crops. So the area under the fodder cultivation is not going to rise much. And there is a specific patterns of this of the growing and the sowing and the harvesting of these fodder crops. So and this the The production of the green fodder requires this rainfall, sunlight, temperature, and so it is all seasonal. So we can see that there are in a complete during the monsoon there is a extra or the surplus quantity of the green fodder available. But there are the two seasons, there are the two period in a year when which is having a very low availability of the green fodder, specifically during the April and May time, and in the November and December time. So we can say that these are the two lean season or the two pockets when the fodder availability is very low. And also the soil fertility and the water resources is depleting, because you can say that because of the encroachments, because of the overgrazing and high pressure on the land, the fertility is decreasing, the resources are depleting. So we cannot say, find that if the livestock is relying on the this fodder grown in the on those field will have a quality fodder. We cannot expect that now. And there is of course there is the feed cost is rising, especially when we talk about the commercial concentrate mixtures. That there is a the protein part, the energy part is having a very high rates these days, and it is very much difficult for the farmer to afford this much of the amount. And likewise, it is also increasing the input cost also. And so this livestock industry need a shift towards a more sustainable, more affordable, and some consistent feed alternatives that not only provide the nutritional stability but that can be stored and used year round. And there should be a very minimum environmental impact and less waste disposal. So the answer lies in the if we are scientifically managing the fodder, or we are going for the conservation of the fodder through the ways of silage and the hay preparation. So why conservation of the green fodder is relevant in this present scenario? Why we should conserve the green fodder and then feed to the animal? Because if we had the conserved green fodder, that can be in the form of a hay or can be in the form of a silage, then we are able to feed the animal during the lean season, which I already discussed that there are the two lean seasons in a year, especially in the tropical Indian conditions, when there is a very less availability of the fodder. And to cope up that, what the farmers are doing, they are just increasing the amount of the concentrate, which is again increasing their input cost. And when we are supplementing the silage or the conserved form of the fodder, then the quality as well as the quantitative fluctuations in the forest supply is minimum, because at this time this silage is having a strict nutritional composition, and the dry matter and the moisture proportions are very fixed. So the quality as well as the quantitative fluctuations that happens when we feed the animal with the green fodder, this there is a reduction in that. And this silage is having a similar composition. The nutritional composition of the silage is fixed, and it is helpful in maintaining the ruminal environment. As we know that these ruminants, the digestion of the feed is solely responsible of the depends upon the ruminal environment in which the microbes resides. And these microbes, they need some substrate as well as a proper ruminal environment to act upon, so that the digestibility can be maximum. So if there is a fluctuation in the diet, if there is a fluctuation in the nutritional composition, then the pH or the osmolarity or the environment of the rumen, the temperature and all, it will fluctuate, which will result in the lower digestibility. So whenever we are offering the animal with a similar composition of the feed, at that time the digestibility remain good, and there is a least impact on these microbes. And for economic feeding of the livestock, as we have already discussed, that silage, it is just alternate for the green fodder. So this green fodder is the most economic form of the feed that we can offer to the animal. So if we are conserving that, then also this is very economic as compared to the concentrate as well as to the straws also. It also enhances the nutrient value of the feeds, as we know that this silage is having a very effective nutritional composition, and it is very much at par to the requirements of the animal as well. And when we are conserving the fodder, So the extra or the surplus water that is available during the monsoon season, because at that time the rainfall, the temperature is suitable for this water cultivation, and so the amount of the water available during the monsoon season is very high. So sometimes it also goes waste. So there is a possibility that you can use that extra one also and can conserve. That can be further utilized during the lean season. It also reduces the content of the anti-nutritional factors. That is because all the feeds, majorly all feeds, contain some or the other anti-nutritional factors. But when we go for the silage preparation, during the process of the ensiling, the concentration of these anti-nutritional factors reduces much, and so this is helpful that the quality of the feed also increases. It also helps in maintaining the ruminal environment because there is a very less fluctuation in the chemical composition, and so the environment or the because there is a very lesser pH fluctuation, the environment is very conducive for the microbial population. And whenever we are going for the fodder, we need to cut it, harvest it daily, and then carry it to the farms and then feed the animals. So it is because of the labor problems and all. If once we had stored the silage and we can daily remove and can feed to the animal directly, so this daily cut and carry system can be discouraged if we are following the silage preparations and all. There is a very less infestation of the worms or the parasite as compared to the when the animal is going on the pastures, because during the insiling process there is anaerobic conditions, and because these eggs or the larva of these parasites, they cannot cope up with the less oxygen or the anaerobic environment, as well as because there is much heat is produced because of the production of the volatile fatty acid, there the pH is low. So because of that, this larva and the eggs of these parasites are not able to cope up with that, and so the infestation reduces. So the aim or the objective of conservation of the green fodder is that we need to conserve the fodder because we need to supplement during the lean season, and we need to manage the extra or the surplus we are having. But the condition should be there that there while conserving or while preserving that, there the loss of the nutrients should be minimal. That is, the nutrient composition should remain nearly as it was when it was fresh. So, coming to the methods of conservation, there are the two common methods of the fodder conservation: the hay and the silage. This can be preserved by drying, whereas the silage, it involves a natural fermentation. As we say, we preserve the fodder in the anaerobic environment. So, what is silage? Silage is a fermented feed which results from the storage of the high moisture crop under the anaerobic condition. Two things are important over here, that it is a fermented feed and the crops are high moisture crops and they are stored under the anaerobic conditions. And it is a green succulent roughage which is conserved more or less in its original form only. So there is a very minimum loss in the nutritional profile, which is the aim of conservation also. And we are anaerobically fermenting this green fodder having the moisture content around 65 to 70%. The green fodder is harvested at a proper stage, it is stored, it is packed, and it is compressed in the silos, and it is then tightly covered so as to prevent the contact of the fresh air. And the process by which this green fodder is transformed to the silage, that means by the process by which this green fodder is stored in a silo so that it is converted into a silage which is a more succulent feed for the livestock, this process is called as ensiling. So this ensiling, that means when we are going for the transformation of the green fodder to a silage, that is the process of ensiling. This ensiling process, it depends upon the moisture content of the fodder crop, how much is the lactic acid bacterial count, because whenever we are going for the ensiling process, there is a production of a lactic acid bacteria. This is the aim of ours, that there should be the higher concentration of lactic acid bacteria, because as soon as there is a high population of lactic acid bacteria, there will be the more acidic environment and the pH will decline. And as soon as the pH will decline, the growth of the organisms that are responsible for its deterioration or spoilage is reduced. And this fodder crop should have a proper substrate, that is a water soluble carbohydrate should be present in these fodder crops, so that they can act as a substrate for these lactic acid bacteria, and then there is a process of fermentation in the anaerobic environment. And the buffering capacity, because what are buffers are the one that resist the change in the pH, but here we want that the pH should decline as soon as possible and as low as possible. So we don't want anything that can resist the change in the pH. So the crops that we want to go that we select for the ensiling process should have a very low buffering capacity. Here, when we talk of the leguminous crop, we know that this leguminous crop, they are having a high content of the protein, but they have a high buffering capacity. So, and they are also not having a high concentration of the water soluble carbohydrates. The concentration of the soluble sugars and the carbohydrates, the soluble carbohydrate is less in the case of a leguminous crops. And these crops are having the high moisture content. Because of the low availability of the water soluble carbohydrates and the soluble sugars in the leguminous crop, as well as because of the high buffering capacity, we do not select the leguminous crop as a substrate for the ensiling. And so we select the crops that are having the high water soluble carbohydrates, that is the cereal fodders are selected for the ensiling process. When they are inside, if we are ensiling this leguminous crop, there will be slow acid production, and there will be extensive degradation of the forage proteins and high production of the ammonia. So it is all loss about. So we never select the leguminous crop for the ensiling process. Rather, the crop suitable for the silage production is always a cereal fodders that are having the high water soluble carbohydrates, high moisture content, and the low buffering capacity. So the principle of ensiling is that involves a conversion of the water soluble carbohydrate into the organic acids. And in this, among these organic acid, we mostly prefer the lactic acid, and this conversion is done by the lactic acid bacteria, which are grown under the anaerobic environment, so as to rapidly reduce the silage pH and inhibit the growth of the microbes or the organism responsible for the spoilage or the decomposition of the nutrients. So, what are the characteristics of the fodder crops that we are selecting for the ensiling? It is that it should have a high concentration of a water soluble carbohydrates, because this water soluble carbohydrate will act as a substrate, and it is required for these lactic acid bacteria to act upon, so as to there is a production of a lactic acid. And these soluble sugars, they are, you can understand, they are high in number in the cereal fodders, but they are poor in the leguminous fodders. And so the for the ensiling, we select the cereal fodders that are high in soluble sugar content. And non-availability of the sugar, if there's a low amount of the sugars, it will delay the fermentation process, because the substrate is not available to the microbes for the conversion, and then the lactic acid bacteria cannot produce much amount of the lactic acid, and it will result in the fermentation losses. So if it happens, then we what we say, we can mix the crop with some other forages, and then we can go for the ensiling of that. And the dry matter, this if there is a if the crop we are selecting is having a high moisture, so the when we are going to compress it, store it, then at that time there is a effluent losses. So on the other hand, if it is a low moisture crop, there the activity of the microbes will be lower. So we need a optimum concentration of the dry matter for the optimum fermentation to occur. So this dry matter should vary between 35 to 50% for the optimum fermentation. And you can also adjust the dry matter content by adding some dry forages or by building the forages in the field itself. So, if we want a rapid and extensive fermentation, we can understand the fodder crop we are selecting should have the high concentration of the water soluble carbohydrates. For example, this can be maize, sorghum, bajra, oat. This can be used for the silage preparation, and the crop should have the low buffering capacity. This is one of the reasons that why we do not select the leguminous crop as a suitable crop for the silage preparation. And the dry matter content should be around 30 to 40%. The levels of the proteins and the minerals should be low because they will enhance the buffer capacity. They will increase the buffering, and there should be adequate concentration of the lactic acid bacteria. Sometimes when it is when this concept we assume that the lactic acid bacteria are low, we sometimes add some inoculants as an additive, so that there is an increase or the speedy production of the lactic acid bacteria, so as to act upon and go for the extensive fermentation. So most important crops which we use for the ensiling is the whole crop maize and other various grasses, whereas we can also use many of the other cereal fodders like sorghum and bajra also. So these are the crops which we use for silage making. These crops are usually having a thick and a solid stems, and they are rich in soluble carbohydrates. And we have also exemplified this the same as maize, sorghum, bajra, and sometimes cultivated and natural grasses can also be inside. But if they are low in the concentration of soluble carbohydrates, we can add some concentration of molasses to it also, and we can also go for mixing some cereal fodders and the legumes in the ratio of 3:1, so as to make it optimum for the silage making. So what is, now we have now till now we have understood that what the silage is and what was the principle behind this, and which type of a crop should be selected for the ensiling process. And so next coming is the silo. That means the structure or the receptacle in which we are going to prepare the silage. It will be called as silo. So it is usually airtight or semi-airtight structure. There are different type of silos, and the types of silos depends on many factors that we will be discussing further. It can be in the form of a pit, it can be in the form of a tar, it can be in the form of a trench, bunker, or there can be some temporary silos made of plastics or the polythene bags. So this picture is showing you the drum silo, the plastic drum silo, the pit silo. This is the bunker silo which we are having in our college, and this one is the bag silos or the poly silos. These are from my lab only. These silos, this bag silo, this drum silo, and this bunker silo we are preparing, and this also, this jumbo bag silos. This also we are preparing regularly over here. So, for preparing these silos, there should be some properties for the site selection. That is, the silo should be approachable from your crop field and from the shed as well, because you need to harvest the crop and then bring it to the silos for the silage preparation, and then from here you have to carry the silage to the shed for feeding it to the animals. So it should be easily approachable from the shed as well as from the crop fields. And the location of the silo is that it is easy to that the silage should be easy to distribute to the animals. We should have some machineries required, as we know that for the silage preparation we will harvest the crop, and then will go for the chaffing of those fodders. So chaffers should be nearby. The area where we are going to prepare these silos should not be low lying, so that that area should not have the waterlogging problem. Otherwise this there is a problem of a seepage, and the silage at the bottom will be spoiled. And it should be at the higher altitude, and in the areas of high water table, silo should be erected on the soil so that there is no water seepage into the silo. And usually it should be away from the milking room, because this silo this the flavor is it gets absorbed in the milk, so it smells. And this selection, this was the slide in which we have discussed that what are the where should we can select the, what should be the points to select the silo. And now we are discussing that which type of silo we want to make, how to select that. It depends upon the number of animals, it how much number of the animals you want to feed the silage, and what is the length of the scarcity period. That means for which period of a time you want to feed the animal the silage, and the kind of the soil, water table, and the drainage system you are having, altitude you are having in your crop, in your the area in which we are going to prepare the silo. What's the depth of the water table, and how is the rainfall and other weather condition over there. These things will decide that how much, which type of a silo will be suitable in your condition. That is, if I'm having a very small amount of the fodder and we are going to feed just to the 3 or 4 animals, then there is no use of preparing the silo pits, long silo pits, large silo pits, that can be maintained in the drums or in the jumbo bags or in the some temporary silos can be used. So we have already discussed that which type of a crops are suitable for the silage making. Then comes when to harvest this. What is the time of harvest of these crops? Because time of harvest is having a major impact on the nutritive value of the silage. If we are early harvesting, then there will be less amount of the dry matter. And if we are late harvesting the crop, then there will be the digestibility will be reduced because of the lignification. Due to the lignin, the digestibility will be poor. So we need a optimum time when we need to harvest these crops, and can that at that time when there is a maximum amount of the digestible nutrients in it, and it is the best time when we are going to transform the green fodder to the silage. That means we are preserving the nutrients over there. So what can be the optimum stage of harvesting the crops for the silage? For the different crops, there is a different proper stage of harvesting. So as for example, if we say of the maize, then it is around 50 to 60 days when there is a from silk to milk stage. And in the sorghum, it is about from 75 to 90 days. Bajra during the oat stage. In the oat, from flowering to milk stage. In the grasses, in the flowering stage, like guinea grasses. And in the Napier Bajra hybrid, when the grass is about 1 meter of the height. So the crops with the thick stems, this slide shows when you will harvest the maize crop for the silage making. So this stage is showing you the milk stage. That is the R3 stage. In this, the grains, what we are getting the grains, they are when you will just pop it down, at that time there will be the oozing of the milk or the white liquid. This is the milk stage. And this one is the dough stage. That means the grains are hard, but still, and the white milk is not oozing out, but it's a firm structure inside. And the milk line should be half to 3/4 down the kernel. So this is the stage when we harvest the maize crop for converting it to the silage. This is the milk line. This one is the milk line. And milk line, very when we you can see that this milk line is varies from 0 to the score of 5. That is when the milk line reaches to the base of the kernel. So when this milk line is halfway down the ground, or it is you can see that this stage when there is a dry matter is around 30 to 35%. In both these stage we can go uh, for the silage preparation. We can harvest the crop at this time, and we can go for the silage preparation, because this time the dry matter content is also optimum, the nutrients are at its maximum, and there is no lignification and no change in the digestibility also. There is some other method also, that how can we go for the analysis of the moisture content in the case of the fodder crops. So this is the you can go for analyzing the moisture content in the fodder crops by using the moisture meter. And the other method is the grab test method, or the fist method that we say. And this moisture meter is not always available to the farmers, and so the other technique is the grab test, in which they can easily analyze that whether this crop has reached to the harvesting stage or not. This is the grab or the fist method, in which in this what we do, we take out the fodder crop and then we go for its staffing, and then hold the whole fodder crop in the hand and try to squeeze out. And if the water is easily squeezing, that means the moisture content is nearly more than 80%. And if the water is just squeezed out and the material is holding the shape, then we can assume that the moisture is around 75 to 80%. But if there is little or no water squeezing out, and but the material is still holding the shape, the moisture content is 70 to 75%. And the other one, when there is no water squeezed out and the is not holding its shape, that means the material falls apart slowly, then the moisture content is around 60 to 70%. And that time we can go for the harvesting of this fodder crop, and we can go for the silage preparation now. So this is the slide that is showing the moisture content when the dry matter content is around 30 to 55. when the moisture is around 70, that means if the moisture content is high, we can go for the wilting of those green fodders. And if the moisture content is very high, you have understood that the dry matter content will be less. And if the moisture content is low, then again what is happening, that the moisture is low, so the dry matter is high, but there is a lignification. So there is a requirement that we should opt for the optimum moisture and the dry matter content for conversion of the green fodder to the lives to the silage. So we can understand, we have understood that when which crop we are going to use, what is the optimum time to harvest that, and what are the types of silo that can be used for the silage preparation, and how this. But and now we are coming to the process of the ensiling. That what are the steps involved in the ensiling process? It is the first we need to we need to select the crop, and then it is to be harvested at the right stage of maturity. Then the fodder is need to be chopped and the chaffed. It has to be loaded into the silo. Then we have to compact and seal so that the extra air is excluded out, and then we can go for the storing. And after waiting for the period of 45 days, we can unload it for the animal feeding. So these are the major steps, and now we will just describe it, that the crop which are suitable for the silage making, that we have already discussed, that these are the cereal fodders that are optimum for the silage making. They are harvested at the right stage of maturity. Then they are chopped to increase the surface area of about 2 to 4 centimeter will be chopping. And then they should be wilted up to the 35% dry matter content. And the silo, the bottom of the silo, they should be this must be free from the moisture and should be above the water table, that we have already discussed when we were discussing that how to what are the basis of the selection of the silo. And it is always advisable to spread a layer of a hay or a straw at the base, so that when we are going to compress the green fodder, the seepages of the effluents and all that can be absorbed in those hay or the straw. And it also prevent the capillary movement of the moisture from the soil into the silo. And if we are adding the silage additives like molasses, they can be added to sometimes if we can find that the fodder is not having enough content of the water soluble carbohydrate, then we can go for addition of these additives. So at that time we need to add this molasses or any other sort of additives, so that there is a efficient bacterial fermentation. And we can also improve the palatability or the nitrogen content by adding some other additives too. Then this chaff fodder is evenly spread throughout the silo and is pressed to expel the maximum air out of it, so as to there is a minimum air in the silo. And this filling of silo must be completed as soon as possible in the shortest period of time, because this when we are filling this silo, this all inside mass is compressed or pressed by the mechanical means or by using the tractors and all, so that there is a maximum expulsion of this air out of the silo. And after filling the complete silo, it is covered with a polythene sheet with a layer of soil or a mud, so that we can make it airtight. And we always prefer to raise the level of the inside mass of a little above the ground, like we make a dome shape, so that the any rainfall, if it is occurring, it should not stay there, it just drop down and just slip off. And the removal of the silos can be done after 45 days of insiling. Then we can open the silo and we can use the silage for feeding of the animals. And if the silo should not be, it should be opened with a very with a great precaution, and this cover should be firmly kept in place. And the minimum face of the silo should be exposed at the time, and then it should be removed out and fed to the animal. This is of some photo gallery. These are the bunkers that we are having in our in my during the my one RKY project I had for the silage, and then we have prepared these bunkers. And these are the two bunkers that we are having. And in this bunker we have first the bottom layer is with a is just having hay or the straw, and then the walls of the bunkers are also covered with the polythene sheet. And then here only the chaff cutter is arranged, and this is the fodder is unloaded, and the chaffing is started, and then this is filling in this is the process when the silage was being filled, when the green fodder is being filled. And this is all about when we are just this is also chaffing of the green fodder is going on. This is when we have filled the silo, and this is in which we are mechanically pressing this green inside a mass, and this green fodder is being pressed with the help of a tractors, and then the polythene sheets are covered, and the layer of a mud is used to make it airtight, and some weights like tires and all can be kept on the surface. This is all how we are making here. This is the another type of the silage we are using. This is a silo pack machine that we are having, and in this we are using this type in which one silo pack is having the 50 nearly 50 kg of the inside mass, and this one is a plastic drum silage, and this one is a jumbo bag silage. It is having an inner polythene and outer is a jute bags. This is the silage, and this all silage we have made here only. These are some photographs that I'm showing the silage to some authorities, and this is a mixing of silage, and we have used some of the unconventional feed also. Some pea waste was also utilized for the silage preparation, and then that was fed to the goats as well. So now coming to the process of ensiling, that how this green fodder is being transformed to the silage. What is the process involved that? So fundamentally, this process starts with reducing the oxygen in the ensiled fodder. That we have already discussed, the silage is a fermented feed which is fermented anaerobically. So the process starts. We have to reduce the oxygen content in the inside fodder. So as there is a the promotion of the anaerobic environment, and the growth of the anaerobic bacteria should enhance. And so these anaerobic bacteria, they act upon the plant sugars and the water soluble carbohydrates and convert them into the lactic acid, or primarily, and also along with the lactic acid, some acetic acid and other organic acids are also produced. So there are usually three phases of the silage fermentation. The first one is aerobic, then comes the anaerobic, and the feed out phase. So in the initial phase of ensiling, that is during the 12 or 24 hours, plant respiration continues and the silo is closed. The cell and the aerobic organism become active, because until all the oxygen is used up, there is a propagation of the aerobic bacteria only. But after this carbohydrates of the plant material is used, and there is a production of carbon dioxide and water, along with the production of some heat, which causes the rise in the temperature of the silo mass, and the air trapped will enhance the production of the Escherichia, Klebsiella, and this phase is having a significant source of diameter loss in the fodder. But this is a mandatory phase. So what we can do, that we need to minimize this phase only, but we cannot remove this phase. So this is the phase one that occurs during the 12 to 24 hours, in which the sugars is being utilized by the oxygen, the aerobic bacteria, and it is converting the sugars to the carbon dioxide, water, and the heat. And because of this heat, there is an increase in the temperature in the inside mass. And because of the utilization of the oxygen by the aerobic organism, there is a decrease in the oxygen content, and there is and when we talk about the microbial growth, there is an increased concentration of the aerobic bacteria. It is the in the day one phase. But when the during day two, the heterofermentation starts. That is, sugars are being converted to the acetic acid as well as to the lactic acid, for more quantity of the lactic acid. That is, it is not much efficient in the terms of lactic acid, but still the some amount of the lactic acid is also produced in the phase two. But in this, the whole oxygen is being exhausted, and there is an increase in the concentration of the acetic acid bacteria. Oxygen is almost constant now, and there is an aerobic environment being set over there, and the temperature again lowers down. And this phase three, it starts from the day 3 to day 6, in which the sugars are converted to the lactic acid, acetic acid, ethanol, mannitol, and carbon dioxide. So again, there is a heterofermentation going on, and gradually there is an increase in the production of a lactic acid bacteria. There is an increased production of a lactic acid, because the population of the lactic acid bacteria is going on. is increasing. And from day 7th onwards, there is a homofermentation in which sugars are being converted to the lactic acid. That is, there is an increased concentration of a lactic acid. This one is more efficient in terms of a lactic acid production, and the concentration of lactic acid bacteria is increasing. So after 30 days, after the day 21, this is all stable. That is the pH conditions, the temperature, and the bacteria population is all constant till the time the silage is again not exposed to the oxygen. So I told you already that the next stage there is a heterofermentation where there is a production of ethanol, lactic acid, carbon dioxide, and acetic acid. And this is not efficient more in this is less efficient in terms of a lactic acid production. And when there is a the remaining oxygen in the inside product is exhausted, there's a transition to the anaerobic phase because all the oxygen is being exhausted. And in the absence of the oxygen, there is a change to more growth by the homofermentative lactic acid forming bacteria. And when this starts, there is a pH will drop rapidly to nearly 3.6, 3.94, nearly this way. And the activity of the lactic acid forming bacteria lasts and the pH continues to drop until most of the sugars and the starch is consumed, and the pH gets nearly, it reaches up to the 4. The level in the drop of the pH depends upon the how much is the concentration of the fermentable carbohydrates and the moisture content of the crop. And once this pH is dropped, the inside crop will be stable as long as the forage is not again exposed to the oxygen. So when the silage is open for feeding to the animals, this again the oxygen accumulation begins gradually, and then the spoilage begins. And as soon as the oxygen intrusion initiates, there is a growth of the yeast and the molds that are responsible for the spoilage of the silage. So what we do that during the when we are opening the silage, the minimum fish should be exposed, and after just taking out the required silage, we need to again cover the silage properly, because if it will the oxygen will intrude, then there is a production of the population of the yeast and the mold will increase, and that will lead to the spoilage of the silage. So there are two types of fermentation. One is a lactic acid type fermentation, other is butyric acid type fermentation. And if the fodder crop is rich in the water soluble carbohydrates, then mostly the lactic acid type of fermentation predominates. But if the forage is rich in proteins, then the butyric acid type fermentation will predominate. And the principal anaerobic microbes, that will destroy the silage quality, it is a clostridia. And this clostridia, this can be classified as a saccharolytic and the proteolytic. And this saccharolytic one will produce the butyric acid, carbon dioxide, and water. But if it is a proteolytic, it will act on the protein part and will produce the ammonia and the amines, which will increase the silage pH. So, and if the acidity rises to about 1% at the start itself, then there is a good quality silage, because at that time this will be the conducive for the growth of the lactic acid type bacteria. And when the acidity is low and the pH is low, at that time when the acidity is high and the pH is low, then the increased population of lactic acid will check the activity of those undesirable organisms. So this is the thing that we need to increase the concentration of a lactic acid growing bacteria. And based on the sugar metabolism, this lactic acid bacteria can be homofermentative and heterofermentative, in which this heterofermentative is less efficient in terms of a lactic acid production. So we have discussed the phases of the silage fermentation. In the phase one, lasts for about nearly 1 or 2 days, in which there is a cell respiration, oxygen is all exhausted, and the aerobic organisms are going on. There is a high temperature, the pH changes from 6.5 to 6, and there is a production of carbon dioxide, water, and the heat. There's a considerable loss of dry matter over this period. And in the next phase, which lasts around 2 to 3 days, there is a production of acetic acid, lactic acid, and the ethanol, and the pH drops down to 5, and the end products are acetic acid and the lactic acid bacteria. And from the 3 days onward, in the phase third, there is a more concentration of lactic acid bacteria, and the pH drops from 5 to 4, and the end products are lactic acid. And from 4 to 21 days, this lactic acid bacteria will be higher in the number, and the pH will be approximately about 4, and the end product will be lactic acid. So how to feed this silage? Now we had discussed the insiling process. What is the what was the what is the chemistry behind this, and how these phases are going on. Now we are coming to the feeding part, that how this silage is to be fed in the dairy herd. So silo size should be decided on the basis of the diameter and the requirement of the forage. So this means, that means number of silos required depends upon the how much animals you want to feed the silage, how much duration, for how much duration you want to feed the animal, how much amount of the silage you are going to feed per day, and which type of, and how much concentration, how much is the availability of the green fodder, is up to you. So we need to see that the 1 cubic meter of the silo can accommodate about 500 to 600 kgs of the silage. So if we are having a 10 animals and we are going to feed them at the rate of 30 kg per day, and for the period of a 5 months, so then about 45 tons of the silage is required to us. So for this 45 tons, and if 1 cubic meter is having a 600 kgs of the silage, then for 45 tons we need approximately 75 meter cube of the space. And this for 75 meter cube, usually the depth is kept as 2.5 meters. This is constant because we do not make the silo height much. We just keep it up to the 2.5 meters. And the width is about 4.5 meters. So the change we can make is with the length. So if it is a 90-meter cube, so we can increase the length up to 9 meters. But if it is a 75-meter cube, so we if we are fixing the width and the depth, so we had a 7.5 meters of the length. So the dimension of the silo can be analyzed if you had the data, that how much animal you want to feed, what is the rate of feeding to you, and how much for how much period you want to feed, and so how much is the availability of the green fodder. So then you can decide the dimension of the that can be a pit or a bunker. And then you understand that when we are converting the green fodder to the silage, there is of course there is some losses in the dry matter. If you assume that there is a 20% loss of the dry matter, so we just if for 45 tons, if the 20% is included, then nearly 6 to 5 kgs of the fodder is required if you want for feeding of the animal for 10 animals at the rate of 30 kg for 5 months. Now, these are the additives. Why these additives are added to the silage? So these silages, when we are adding to the, when we are adding any additive to the silage, suppose there can be various purpose of adding these additives to the silage, because they can improve, they can be used as a to enhance the insiling process, to speed up the insiling process, to reduce the losses, to reduce the aerobic deterioration. That means when we are using it for the feed out, at that time I already discussed that is a aerobic deterioration of the silage. To reduce that, we can use silage additives. To improve the hygienic quality of the silage, we can use additives. To limit the secondary fermentation, because the secondary fermentation will lead to the losses in the dry matter. So to limit that, to improve the aerobic stability of that, to enhance the nutritive value of the silage, we can use the silage additives. And there are various kind of silage additives that can be used for the better silage preparation and for improving the silage quality. These silage additives, they are added at the time of a silage preparation, and they aid in the better silage fermentation and reduce the fermentation loss. These additives can be fermentation stimulants like microbial inoculants, in some enzymes or the sugars, and they can be fermentation inhibitors like some acids like formic acid, lactic acid, mineral acid. They can be some chemicals also. There are some additives in the category of that can inhibit the aerobic deterioration, like inoculants, propionic acid, acetic acid. They improve the aerobic stability. Some nutrients to enhance the nutritive value of the silage. We can add urea, ammonia, minerals, and some grains, and also that the nutritive value is also enhanced. We can also use it as an adsorbents, because so that the extra moisture can be retained. So fermentation, so usually we use the fermentation stimulant as the silage additive that is more commonly used, and many of them are available commercially. and naturally lactic acid bacteria on the inside crop would be sufficient to ensure the satisfactory fermentation. But sometimes when these crops are having a poor concentration of the lactic acid bacteria, then we can use some of the commercial microbial inoculants. These are the dried cultures of the homofermentative or the heterofermentative lactic acid bacteria. We can use at a particular dose rate that is given in the in that booklets and all. So commercially we can use them also, and then we can speed up the fermentation process. And so now when the silage is prepared, the concern is that what is the silage quality? How you can assure that the silage you have prepared is of the good quality. So it is usually we determine it by the odor, its physical state, its pH, ammonia nitrogen concentration, and the volatile acid and the lactic acid concentration. So physically just we see the smell and the odor and the texture of that. It should be I'll be just describing in the subsequent slides also. So silage quality parameters, it should be acidic, the pH should be nearly 3.99 or nearly 4. Ammonia nitrogen should be 7.87 to 10% only. Lactic acid concentration should be above 3 to 5%. Acetic acid concentration should be only up to 5%, and the butyric acid concentration should be 0 or less than 0.2%. So these are the parameters which we use for the silage quality determination. And as we see the color of it, it should be preferably light green to green brown. It is a normal color range for the cereals and the maize silage. And if it is a dark brown, if it is a straw yellow, if it is a very dark or a olive green, so it all says that the silage you have prepared is not of the optimum quality. And sometimes when there is a high temperature also, then also this silage converts to the dark brown color. It is because of the extensive heating, and there's a significant loss in the digestibility and the protein portion, and so this is not preferred. High temperature also gives you the blackish and brownish black type of a color. And when the temperature is moderate, the silage tends to be yellowish or brownish greenish, and sometimes it is turns to be golden in color because of the action of the organic acids, that means the acetic acid or acetic acid, on the chlorophyll pigment that converts the chlorophyll into the brown magnesium free pigment that we say as a phyophytin. So usually the color is preferable color is light green to greenish brown or the golden yellow color. And when we evaluate we also evaluate it with the odor, and if it is smelling like a vinegar, so that definitely it means that the concentration of acetic acid is high. That means you have gone for a slow filling of the silo, or if your green order was too wet. And if that, or the reason can be that it was not pegged or sealed properly. And if it is smelling like alcohol, that means there is a production of a ethanol. So definitely the some oxygen has entered, and there is a yeast fermentation is occurring. If it is smelling sweet, so that means it is okay. And if it is a rancid smell is there, that means there is a clostridium growth is there, and that is the most that is the, this one is the principal bacteria which is responsible for the deterioration of the silage quality, and there's a high concentration of a butyric acid. If it is little smelling pleasant, that means there is a lactic acid bacteria. Lactic acid is produced in an optimum quantity, and this is the preferred smell of the silage. And if it is a tobacco smell or a caramel smell, it is a heat damaged one, which is not preferred. So this can be categorized as, if it is a very good silage, it should be acidic taste and odor, and it should not have any traces of the butyric acid, molds, or the sliminess. The pH can be around 3.5 to 4.2. Ammonical nitrogen should be less than 10% of the total nitrogen, and lactic acid concentration 1 to 2%. If it is a good silage, it should be again acidic taste and odor, and traces of butyric acid only 0.2%. pH can be around 4 to 4.5, and ammonical nitrogen 10 to 15%. And if it is a fair silage, there are subbutyric acid concentration, molds are present over there. Some sort of proteolysis has also taken place, which is converting degrading the proteins to the ammonia, and because of the presence of the ammonia, the pH is high and 4.8 and above, and because of the production of the ammonia, the ammonical nitrogen concentration is also high. So this will come under the category of the fair silage. Usually the silage should fall into the category of the very good silage or the good silage, considering these parameters. Yeast are generally the initiators of the as soon as the air will enter, the yeast and the mold growth will increase, and this will consume the sugars and the fermentation acids, and it will raise the silage temperatures, and also the pH will be raised. So, and with the increasing pH, bacilli and other aerobic bacteria will initiate, and it will completely destroy the silage. So some undesirable microbes like Clostridia, usually they are present on the crops because this is present in the soil, and in with the green when we harvest the green fodders, this soil is also the contamination of the soil also occurs in the form of the spores, and there are many spores of the Clostridia in this soil, and they also when they reach the when they are when the inhaling starts, they grow under the anaerobic condition also. And we have already discussed that these Clostridia bacteria, they can be saccharolytic as well as a proteolytic, and this saccharolytic will act upon the water soluble carbohydrate, and then they will produce the butyric acid, which will increase the pH. And we have already discussed that the increased concentration of the butyric acid is not preferred as a good quality silage. The high concentration of the butyric acid will not be considered good for the silage quality. And if there is a proteolysis is occurring, that means your protein is being degraded to the ammonia, and it is going wasted. And then there's a conversion to acetic acid in bacteria, amines and ammonia. Because of the presence of the ammonia, there will be the high pH. So this is not preferred. So we should avoid this the production of these undesirable microbes. The solution is that there should be increased quantity of the lactic acid bacteria. As soon as the acidity will enhance and the pH will drop down, the aerobic bacteria or the growth of the undesirable microbes will be checked down. But still, there are certain losses during the insiling process. The major one is the field loss or the pre-insiling loss, that is insiling that is losses before the insiling occurring in the field, or the insiling losses and the effluent losses. Next. And the field losses, if we are harvesting the crop and on the same day we are going for its staffing and the inselling process, then there is a minimum losses. But if you are wilting it, you have seen that the moisture is high, and you have thought for wilting that green fodder in the field itself, and if you are wilting it for more than 48 hours, there is a considerable loss of the major nutrients. And when we are cutting the fodder crops, there is aerobic fermentation will start, and that means this aerobic bacteria, they will use the oxygen, and the substrate of that means the water soluble carbohydrate will be utilized by them, and they will be converting into the carbon dioxide and the water and the heat. So again, silo must be filled within the 24 hours of the harvest so as to avoid the pre-insiling losses. And the other one is oxidation loss. We have already discussed that because when we chaff the fodder, the respiration still continues because oxygen is still available there. And when this oxygen, aerobic bacteria will utilize this oxygen and will convert the water soluble carbohydrate to the carbon dioxide, water and the heat. There is increase in the temperature of the silo, and if you are not compacting it properly, their air pockets are still present, so it will again lead to the much of the aerobic losses. And this loss can be minimized if you will properly compact the inside mass, because then the maximum quantity of the air is extruded out. And in the anaerobic losses, due to the formation of the volatile compounds like alcohols, amines, carbon dioxide, extra, this is all losses because of the high number of the heterofermentation. Because during the heterofermentation, there is production of ethanol, mannitol, carbon dioxide, acetic acid, and this loss can be account nearly to 4 to 6%. And sometimes the other one is the effluent losses. When we are compressing the inside mass, the are also carrying with them the soluble nutrients. And this can be prevented if we are keeping the moisture content up to the optimum quantity. If there's a high moisture in the forage, then the effluent losses will be much, and along with that effluents, there will be a loss of the soluble nutrients with that. So these losses can be, we can say, during the respiration, 1 to 2% of the loss is there, and but this respiration losses are inevitable, and for the fermentation also there are 2 to 4% losses. It is also inevitable. Effluent losses, these are also inevitable. But we can prevent the secondary fermentation and the losses because of the aerobic deterioration during the storage time and the aerobic deterioration when we are discharging the silage. So these can be prevented. That means the losses, some losses are inevitable. So we can minimize that, but we cannot completely wipe off that. So, but certain losses can be preventable, like secondary fermentation, aerobic deterioration during the time of the storage, and aerobic deterioration during the time of the discharge of the storage, silage. So now coming to the benefits of silage making, we have seen that the nutrients are preserved here because we are harvesting it at the time when there is a maximum concentration of the nutrients, when the total digestible nutrients are its maximum. That means when the biomass is also optimum, when the dry matter is also optimum, when there is not the lignified mass is there, or when there is a high content of moisture is there, we are selecting a right optimum stage of maturity, and at the time we are harvesting, so we are conserving or preserving the nutrients in that. It is also more palatable and succulent. It enhances the digestibility. There is a lesser moisture variation in the diet because here the moisture content is 60 to 70% and it is fixed. But whereas when we are feeding the green fodder, sometimes it is 10%, sometimes it is 20% dry matter. Moisture content varies with the season and the stage also, and it is less weather dependent. Previous slides. It is less weather dependent. It can be done manually. We can prepare it manually or using or mechanically also. It can be utilized for both the type of farms like backyard farms or the commercial farms. We can use a wide range of the crops. We can mix and merge two, three types of fodder crops. This feeding can be sustained for a longer period of a time, and compared to hay also, there is a less nutrient losses over here because when we are preparing the hay, there is a 30%, nearly 30% of the dry matter is lost, but when we are preparing the silage, the losses are less. And but it is also of course more economical, and if we can use a land because we are harvesting it at a certain stage of maturity, so your field are again vacated for the next crop, so you can use a land with 3 or 4 crops annually, and it requires less time for less space for the storage as compared to the hay. It is cost effective and sustainable way to provide the herd with many of the nutrients and calories, and we can keep our livestock healthy and more productive. We can also prepare the total mixed ration with that, and it can be supplied throughout the year. Like we have seen that there is a reduction in the concentration of the anti-nutritional factors when we are going for the ensiling. We can see that when we are ensiling it, there is a 23% reduction in the nitrate concentration in the case of guinea grass, and in the 62% of the oxalate will be reduced during the ensiling in bajra, and in the Napier bajra also there is a reduction in the oxalate concentration by 54%. So you can see while ensiling there is a reduction in the anti-nutritional factors of the fodder crops. And when we are comparing the nutritional composition of a green fodder and the maize silage, we can see that the dry matter is enhanced, the protein content is enhanced, the fat content is enhanced, the lignin concentration has reduced, the total digestible nutrients is increased, and toxins are reduced. Buffering action is there. So with this, we can say that at least we can, and there is a lesser variation in the diet composition when we are using there is no variation in the diet when we are using the silage as compared to the green fodder. So we can see that the nutritional profile is also good when we are using the silage. As we have so many times we have discussed that there is a similar feeding throughout the year. Suppose I am giving you the example from January, we are usually having here the persimmon and the oat, and we are giving it 20 kgs at that time because there is a availability of the persimmon and oat, and with that we are giving 3 to 4 kg of the straw and 4 kg of the concentrate mix. This is the traditional type of a feeding in which we are not using the silage. But other group, if we are going for the silage feeding, we can say we have fixed the silage quantity as 15 kg, and straw 2 kg and 3 kg concentrate mixture. During the February month, there is still the persimmon oat is available, and we are giving similar feed, that is 20 kg persimmon oat, straw 3 to 4 kg, and 4 kg of concentrate mixture. But when coming to the March, the persimmon oat availability is reduces. So to compensate that, what we do, we have increased the concentration of the dry and as well as the concentrate and the amount. We have increased the amount of the straw as well as the concentrate. During the April season, Basim and oat are not available, or some amount of Basim is available along with the napier. And then also we have increased the concentration of the uh, concentrate measure. But here you can see we have just kept the group which we are keeping on the silage feeding. We are just giving a constant value of the silage dry and the concentrate measure. So we can see there is a variation in the feed type also, and variation in the nutritional profiling also, because the bursting in the January is not the same bursting in the March. There is a lot of variation in the dry matter and the moisture content. So there is a variation when we are going towards the when we are feeding the animal with the traditional feeding, and then there is a variation in the feed. And but with the silage feeding, the composition stays similar throughout the year. But there are certain limitations also. It requires some machinery like chaff cutter. It also requires some tractors and the shed and the preparation of the silos should be there. And you need to repair the silo sometimes, and you cannot preserve the high leguminous foders. You cannot use those foders for the silage making, and the loss of the nutrients can be high if you are not properly excluding the air. Plus, sometimes secondary fermentation are taking place. Sometimes there is a aerobic spoilage is occurring. Seepage losses are there. Effluent losses are there. Ensiling losses are there. So this can be and some and this. Of course, the silage preparation needs some technical expertise and the advice of some scientific person, or some training in handling should be given before you go for the silage preparation. So these are some of the limitations of the silage making. These are the photographs how we are making the silage in our college. This we have already discussed.